Hey everybody! Today we're going to be talking about the pour plate technique that we're going to be using in class. And the pour plate technique is one way for us to sort of do what we call plate counting, a concept that will be covered later on in the semester. But the pour plate technique, it is sort of a test used to look at the quantitative testing for the presence of microbes in things like food and beverage items. And because of that, samples generally have to be diluted multiple times through a process called the serial dilution, another concept that we'll cover later on. But when it comes to the pour plate compared to what we call a spread plate, the pour plate allows for growth both within the medium and on top of the medium, whereas the spread plate only allows for growth at the top. So there are different applications for different types of testing that can be done with these different techniques. But you should know again that the pour plate allows for growth both inside and on top of the medium. Now let's discuss the different materials that you're going to need in order to perform this technique. So you will need a Bunsen burner and a striker. You're going to be needing your sample, whatever sample again it is that you're testing, and this has to be a liquid sample. So in this case here again, I have a broth here. If this was like a food item, you would have to kind of break it down into a liquid food item, like mush it up in a blender. And of course, if it's a liquid, you just test the liquid directly. But overall, it has to be a liquid sample that we use, so a broth for our class. You're also going to need a pipetter and a pipette in order to do your broth transfer. And then you're going to be needing a sterile petri dish, basically for you to put your sample into. Now the last thing that you're going to need is then going to be a boiled agar deep. And the thing here is that our boiled agar deep is now a liquid. And normally what we do is we kind of have a setup like I have here on the other side of my table. And we have this water bath that keeps our agar at about 45 degrees Celsius. And the big thing here is that at that 45 degrees Celsius, the agar stays liquid. However, it is also cool enough that once we add it to our sample, it should not kill the bacteria in there. But once it gets down to about 42 degrees, it is going to start solidifying, so we have to be kind of quick with the last step of our technique. But let's go ahead and let's kind of talk about the procedure for a pour plate. So let's go ahead and get our Bunsen burner going. We'll kind of take it from there. All right. So again, my overall goal, I want to get my sample from here into there, then I want to pour my agar deep on it, so I'm basically making my own plate. And then I'm going to be looking at where do these different microbes grow in. So I'm going to take my disposable pipette, I'm going to break open the end with the cotton swab in it. I'm going to insert it into my pipetter. I'm going to take my sample, uncap my sample, pass the mouth through the flame, dip my pipette into the sample, and I'm going to pull a full ml out of it. Remove the pipette, flame the mouth of the sample tube, and recap it. And now what I'm going to do, and this is very important, you have to put the sample down inside of the base of it. So I'm just going to release the pressure. I'm going to put it right in the middle of my plate. Push the top in order to get the rest of my sample out. Close the lid. I'm going to go ahead and put this back in the bag, put the pipette back in the bag, and then we can go ahead and biohazard our pipette. Now again, I mentioned it is very important that we put this down in the bottom of it and that we do not put it on the lid. And the big thing here is that the bottom holds quite a bit more media than the lid does. So when we're going to be pouring our stuff into it here in a minute, if you put your stuff in the lid and then try to pour that whole agar deep in it, it's going to go everywhere and you're going to make a huge mess basically. So got to make sure again that we try to get it basically in that bottom. But that does kind of get us into our last step. I'm going to go ahead, I'm just going to grab one of these, which is my boiled agar deeps. I'm just going to hold them in my hands just for a little bit, basically cools them down just another degree or so. And once I've done that, uncap your agar deep, open the lid, and then kind of pour it gently into the top of it. And now I have basically just made my own little plate. So the tube can just go into the biohazard after that. But now the big thing here is that I do not want to touch my plate for the next 10 to 15 minutes. And it's about that long that it takes for my auger to actually solidify. So if I start moving it around, and especially if I try to flip it upside down, that auger is going to go everywhere. We kind of have to give it again that 10 to 15 minutes so that we can actually wait for the plate to form itself. And then after that, we just put it in the incubator so that we can look at it during our next class. But really, that should cover everything that I need to talk about in terms of the pour plate technique.